Hello, everyone. Today we're going to talk about how cells divide, specifically mitosis. And what I want you to know first is that there are two different categories of cells. So as it says here, there are sex cells or there are body cells. Sex cells are also called gametes. Sometimes you'll hear them referred to as egg or sperm. And they are only used in reproduction. Lastly, they only contain half of your genetic information. Second type of cell are called body cells. Those are also sometimes called somatic cells, and those are any kind of cell that isn't a sex cell. Those cells contain a full set of your genetic information. So, for example, in a human, there are 46 chromosomes in a normal human cell. So, in a body cell, if it has a full set, then it has all 46 chromosomes. In a sex cell, because it only has half of your genetic information, it would have, instead of 46, it would have 23 of the chromosomes. When cells divide and make new cells, there are two different ways that happens. One is mitosis, which is going to be our focus today, and the other one is meiosis that we'll be talking about in a later lesson. So our body always needs new cells. And like I said, there are two ways that it does it. In mitosis, this is when we want to make new cells that help us grow. Or sometimes we have old cells that we need to replace or repair. All right, so that for that purpose, we will use mitosis. Meiosis is when we want to make sex cells for reproduction. Though that process is slightly different because we need to end up making cells that only have half of our genetic material. So to talk about mitosis, what we first need to understand is that a cell has a life cycle, just like when you learn in elementary school about butterflies and they go through their different stages of development. There's the pupa, there's metamorphosis, and then you end up with a butterfly, even though you started as a caterpillar. It has all these different stages. Your cells have different stages of life as well. And so there's G1, there's S, G2, and M. During G1, a cell grows and does its normal job. During S, it starts to get ready for division, and the first step of that is to make extra copies of the DNA. Because if I'm going to divide, I'm going to need enough DNA for two cells, not just the one I already have. So that is the synthesis stage where we replicate DNA. How replication happens, we will talk about in our next scenario. In G2, the cell continues to do its regular job, but it also starts to continue to prepare for cell division. So we already copied the DNA in the last phase. So in this phase, it's making copies of all the other cell parts. So things like the ribosomes, the endoplasmic reticulum, the Golgi apparatus. I got to have all the parts to make two complete cells. So at the completion of G2, I now have all the parts that I need to make two new cells. So at that point, when it's ready, it will go into the mitotic phase. This is where things actually separate into two cells. That mitotic phase is when mitosis occurs. All of this doing its job and preparing for that is called interphase. The cell doesn't really look like a whole lot's going on if you look under the microscope. Nothing really starts to look different until we get into the M phase. So we're going to focus our conversation today on the M phase of mitosis. Again, remember that the purpose of mitosis is to help us grow. All right, so we need to make more cells. Think about the idea that you started as only one cell big when the egg and sperm came together, and now you're over 1 trillion cells. So where did all those cells come from? Well, mitosis, the cells divided. And also, like we said, to repair old cells that are just need to be replaced. They don't work well anymore. They've been damaged. So almost all of your body cells, or somatic cells, all right, vocabulary word here, complete the cell cycle and undergo mitosis. Somatic cells are any cell, again, that are not a sex cell. So in other words, anything that's not an egg or sperm. So skin cells, kidney cells, heart cells, all of those cells are going through the cell cycle. The only exception to that are your nerve cells. Neurons do not go through the entire cell cycle. They stay in a permanent interphase. So after G2, they do not undergo mitosis. They go into a place called G0 or G sub naught, and they stay there. Why is that important? Well, I want you to consider the fact that that means that those cells do not continue to make new ones. So if you damage them or they stop working, you can't fix it by making new ones like you can with your skin or heart or liver or kidney cells. You know, when you cut your skin, your body fixes it by making new skin cells. When your brain cells are damaged or die, they cannot be replaced. So what kinds of things damage brain cells? Well, drug use, blows to the head. So, if you fall and hit your head, or, things that cause things like concussions, for example, those kill brain cells. And so you only have so many brain cells. So if you continue to, kill brain cells over a lifetime, eventually you're going to see some kind of effect. So I'll give you an example. In the case of Muhammad Ali got Parkinson's disease, which is caused by cell death in an area of the brain that controls motor function, which is how your body moves and does things. So he got it at a relatively young age. So why? We expect there to be a lot of brain cells gone by the time you're in your 70s, 80s, 90s. You've been accumulating dead brain cells over the course of your lifetime because your brain doesn't fix and replace them. But at a young age, we don't expect you to have a lot of dead brain cells in certain areas of your brain. So why did Muhammad Ali get Parkinson's at such a young age? Well, think about Muhammad Ali's job. He was a boxer. He continually got hit in the head. And so all of those blows to the head killed brain cells over time. This understanding of brain cells not coming back and causing brain damage over time was the basis of a lawsuit against the NFL. The athletes know that they continually get hit and there are brain issues from all of those blows to the head. So there's all kinds of repercussions of prolonged blows to the head or drug use that kills brain cells. Those cells don't come back, and it can lead to major disease, because when those cells are gone, it affects certain areas of the brain. So let's talk specifically about how do we make those cells? How do we make new cells when we need them to grow or replace old damaged cells? Mitosis is a process of cell division that ends up making two identical daughter cells. Why do they call them daughter cells and not son cells? I don't know the answer to that, but they're called daughter cells. And it is important to note that they are identical. When I cut my skin and want to make new skin cells, I don't want to make a heart cell. I don't want to make a liver cell. I want to make an identical copy of what was there before. There are 4 parts to mitosis: prophase, metaphase, anaphase, and telophase. And that's the order they happen in. Sometimes students like to remember PMAT to remind themselves what order. After the nucleus is divided, the cell actually separates into 2, and that process is called cytokinesis. Sometimes you'll see that included as a step of mitosis. Some books put it in with the 4 steps and make it 5. Some put telophase and cytokinesis together and keep it at 4. It just depends on the book that you read. So in each step, something different is happening in each phase of mitosis. So on this slide here, you can see a bunch of cells. The red are the nuclei of the cells. And so when a cell is in prophase, the DNA tightens up and coils into these thick pieces. So, for example, over here, you'll notice you don't really see anything inside the cell. And in those cells, mitosis, those isn't really happening yet. It's in interphase. You can't really see anything. Even these, you can see the red nucleus, but you don't really see anything in there. It just looks like a red blob. So when mitosis is starting to occur and the DNA thickens and condenses into chromosomes, you can actually see the strands. So you can see here it's starting to happen. So this is early prophase. And over here you can actually see the strands a little bit. That would be what we call late prophase. And also the nuclear envelope starts to disappear. So the nucleus has a bit of a shell around it. It's called the nuclear envelope. And so for a cell to divide, we got to kind of let the DNA move and spread out. And so that shell around the nucleus dissolves, and you don't see a defined circle anymore like you do in these other ones. It disappears, and then the DNA can spread out. In metaphase, the chromosomes align. They line up in the middle. All right, and so you see that here. They're all lining up here in the center of this cell, and spindle fibers, kind of, I like to call them like spider webs, like from Spider-Man, and they come from the opposite sides of the cell, and they kind of spread out and attach to all the different fibers of DNA. All right, and something called the centromeres, which is where the spindle fibers are coming from. You can't really see it in this picture, but they are where the spindle fibers come from. Those remain intact. In anaphase, those spindle fibers start pulling the chromosomes apart. And here you can see that in this slide. So you can see that they used to be in the last slide all lined up in the middle. Now they're pulling apart to different sides. When you think about making two new cells, I want to make sure that the DNA gets separated evenly and properly, that I have all the right genes in the right cells. So that's why they line up, and then they separate or are pulled to opposite sides so that each side that will eventually become a new cell has all the DNA that it needs. Once everything is separated on the inside, then telophase begins. And in telophase, the chromosomes have fully separated. They're all the way on separate sides. And the cell starts to pinch off. You see it just beginning to pinch into two. In cytokinesis, the cell completely separates. So it totally, you see the cell membrane develop in between here. The nucleus of the two daughter cells form. Right? Remember that our two cells at the end are called daughter cells. And the DNA starts to relax and unwind, and it moves back into interphase again. So again, that center nucleus, you would start to not see the DNA so much because it unwraps. It's not so tight anymore. All right, so how would we represent this if I were talking about it? This is very specific, so we're looking at actual slides of cells. But what are some other ways we might talk about it? All right, so to summarize mitosis, I want you to remember that we start with one cell. Let me get my marker here. We start with one cell, and that one cell is going to divide and make two cells. All right, so some things to point out. All right, so for example, this cell up here is our original parent cell, right? That's what we started with. The two down here are our daughter cells. And if we recall, those cells are identical copies of the parent cell. All right, so how do we apply that? So if I told you, for example, that we had a body cell that was human. So human cells have 46 chromosomes. And I told you that this particular body cell was going to undergo mitosis. And I said to you in a question, let's say on a test or quiz, I said to you, how many chromosomes would be in the daughter cells? Well, I can answer that because I know in mitosis that when the parent cell divides and becomes daughter cells, that they are identical copies. So that means that these daughter cells have to have 46 chromosomes as well. Hopefully that helps you better understand mitosis. In our class discussion, we'll be able to talk about the importance of mitosis and how it relates to cancer.